Well, hello again, and we are going to continue our discussion about the nucleus and endomembrane system. And where we left off with the last video, we were looking at the nucleus, and we're going to start now with the ribosomes. Now remember, what we're going to be looking at are the organelles involved in this very unique assembly line, and then we're going to talk a little bit about the ramifications, the applications, the type of cells that would be using this, and some examples. So let's switch to the ribosomes. The ribosomes are going to be found in two places. So we have free-floating ribosomes, and that means they're going to be within the cytoplasm. Now, and just a quick note, I'm going to go over these two, but a ribosome could be free-floating, and then they could attach to the rough ER, and then vice versa. So they don't have a permanent position, but where they are does dictate what kind of proteins they make. So free-floating ribosomes that are in the cytoplasm, they're going to be making proteins that function in the cytoplasm. I can't write tonight. So they are going to be making proteins functioning in the cytoplasm. So a good example of this might be an enzyme that is involved in the metabolism of glucose. And so as glucose enters the cell, it's going to be within the cytoplasm, and so you could have an enzyme right there that is going to add or join something to the glucose or start breaking it apart, and then that would be from a free-floating ribosome. Now the second one is, from my perspective, a little bit more interesting. You have ribosomes that are bound to the rough ER, the rough endoplasmic reticulum, and you can see these guys all along the rough ER right here. All of these, they look like little grains of pepper. Those are the ribosomes. And the line that you see right here, that is a membrane from the rough ER, and so is that one, and then you see another one down here, and then another one right in here. What will happen, and I might have to zoom in on this so you can see, or I can draw something in it. These guys are going to be making proteins, and these proteins are going to go inside the lumen of the rough ER. So just on this electron microscope picture, the yellow would be the proteins. This is going to be highly important because that's going to give us an idea of what kind of proteins and what these proteins do. So there are three pathways that these proteins can take. One, these proteins can be destined for secretion. So these proteins will be secreted outside of the cell. And the reason they have to then go into the rough ER is that's the way to get it outside of the cell. And you'll see that as we move on. The second one will be proteins that are for the membrane, that are embedded within the membrane. And then the third option is going to be proteins that are within. Now the key there is within. It's inside other organelles. So we're going to take a look at these three places. Now to start that, then we have the rough ER. So the rough ER is this complex sheet of membranes. All of these are membranes, and you'll notice that they are continuous with the outside of the nucleus. So let me just draw this line in, and you can see that membrane folds back and creates this complex system of tubes. And then here's another one. And notice that this joins to make that next layer. So you can almost imagine, let me get my yellow again, you can almost imagine a protein that's made within here could then travel throughout this system of tubes. And as it goes from system to system, and of course this is just a cutaway, so it could eventually get up here and the proteins could gather into this little vesicle and then pinch off to go somewhere. And that's one of the keys to understanding this whole assembly line. So what does the rough ER do? Well, it can serve as a transport, a means to take a protein to a certain spot. But oftentimes, you're going to see enzymes within the rough ER that modify proteins that modify the proteins that are made in there. Usually what this involves is adding carbohydrate groups to the protein. And these were from a previous video that we watched. These might be oligosaccharides, those small chains. They're bigger than disaccharides, but they're not huge polysaccharides. And so you might have, let me go ahead and draw this, you might have a nice big protein, globular protein, that then has these little carbohydrate chains added to it. And because of the carbohydrate and the protein, we call those, we call those glycoproteins. And you can also do the same thing with lipids, lipoproteins. And you don't even need to necessarily add anything. It could just change the type of protein. Maybe another polypeptide is added to it. Maybe you cleave a part off. Maybe you break something off of it. So that's the rough ER, and then it's going to ship, and it's going to ship this most likely to the Golgi apparatus. Now, just to show you an electron microscope image, over on the right here, we see the rough ER with all of the ribosomes sprinkled throughout this on their membrane. Over on the left, we're seeing the smooth ER. Now, the smooth ER is not involved in the protein manufacturing. It does have very important jobs, though. It can synthesize important lipids. So it's involved in the synthesis of lipids. One important example of this would be steroids. So any part of an organism's body that would be making steroids, like estrogen, testosterone, or the adrenal gland steroids, would have a lot of these smooth ERs. They can also be involved in detoxification. Detox of like alcohols and other toxins, poisons. Depending on the dosage, the liver can handle that. And what's interesting, this is a nice application. If you have a toxin, what the smoothie R can do, they will add these hydroxyl groups to it. Now, why is this so interesting? Well, what this does is it makes it more soluble in the fluids, and then the animal or our bodies can then flush it out. It'll be removed much easier. So it's an interesting chemistry application. It can be involved, the smoothie R can be involved in carbohydrate metabolism, also calcium storage in the case of muscles. So we'll get to that in specific examples, but that's kind of an interesting sideline right now. And then we get to the Golgi body, and the Golgi body continues our study of this assembly line. So just to kind of review, we started with our ribosomes, and we talked about the ones that are bound to the rough ER. And so keep in mind, these ribosomes are making proteins that could be secreted outside the cell, proteins that are embedded within the membrane, and then proteins that are within other organelles. So here we're going to really finish that up. Now the Golgi body is where the last step is. So what happens in the Golgi body, you have further modifications to the proteins. And so again, you can get carbohydrates, lipids, you can get these attached to the proteins. And let me use my yellow again. So what can happen is um, not only is it modifying these guys, but it's going to collect them. And once it collects them, it helps tag the location. So we see these molecular labels on these non-located locations. Um, and that's going to be really important because now the Golgi body becomes a shipping and packaging center. And I know you've probably heard in previous classes us talking about it kind of being a UPS or a FedEx or the post office. And so once it gathers those proteins, they can pinch off and then go to their respective places. Now, the easiest idea to think about is this could then become a lysosome. This little bundle. It may not be mature yet, but that's maybe a more intuitive sense. Once we've got this little vesicle, whatever proteins are within it, these proteins could then go off and perform their job. So this could become a lysosome. So what are the other two possibilities? Well, let's just imagine now, let's just take the example of a membrane protein. And let's use red. And so if it's a membrane protein, you want to keep it in the membrane. So even as far back as the rough ER, this membrane protein, let's try to get a thinner one. This membrane protein is going to be part of the system. Can I get a thinner one? There we go. And so if we have these membranes that are embedded in, then let's take receptors, and these receptors are going to have a sidedness to them. And I'm going to use that as an example because that's very important, because how they exist on the outside of the membrane dictates how they're going to appear within the vesicle. So these membrane proteins are going to travel embedded in the rough ER membrane and then embedded in the Golgi body membrane. And then if we put it all together, let's go ahead. Let me zoom in on this. So let's take the example of our membrane protein. So if these membrane proteins, if they are receptors that have a sidedness to it, in other words, one side is different from the other, that sidedness has to continue on. So let's just keep going with tracking this membrane protein. And so what we're going to do is we're going to start gathering them together, embedded in these little vesicles, and you're going to see exactly what happens. This little protein, these proteins that are embedded in the membrane, watch what happens. That membrane fuses with the cell membrane, and all of these proteins that were facing the inside of the Golgi body, as they flip and invert on the cell membrane, these are now facing on the outside. So these could be receptors. These could be glycoproteins that identify the cell. And then over here you see a vesicle that could become the lysosome. We already talked about that. Or in this case, you see the molecules that are being secreted. So we're going to take a look at some examples over the next couple of days, but now you see the factory line. So these could either go and stay within the membrane, they could go outside the cell and be secreted, or they could stay in the cell but be collected to form another type of organelle. So we'll look at some examples tomorrow in class. Thank you for watching.